Hello, I'm Deshaun Yeke, and this is my presentation. So, to start off with the cell cycle, you can see that there's two phases, interphase and mitosis. And if you look at the diagram, you can see that there's many subphases between these two. In interphase, you have G1, S, and G2, which is all based on growing the cell up and doubling the chromosomes. And mitosis, as we all know, has prophase, metaphase, anaphase, and telophase, which splits up the doubled chromosomes into two. Now this sounds like a lot of work, but the cell cycle is being able to regulate it because of its proteins and the job it does and the checkpoints and how it only allows healthy cells to go past and go through the process. So the first checkpoint we have is G1 checkpoint, which is in the G1 phase, and this is just the beginning. This is like a security check where they make sure that the chromosomes are doubled, and if they are and they meet requirements, they get to go past, go through all the phases, and most of them go and complete mitosis. But if they do not have the requirements of doubled chromosomes, they get sent to the G0 phase, which is cells that cannot be divided. In the G2 phase, or the G2 checkpoint, is at the end of the G2 phase and almost close to the beginning of mitosis. And this is just like a secondary checkpoint. It's making sure, double checking that all the chromosomes are correct, all the chromosomes are doubled, so that processes in mitosis are easier. And in mitosis, there's M phase, M for mitosis, and it is between the metaphase or subphase of metaphase and anaphase. Metaphase lines up all the chromosomes into a line down the center of the cell. Anaphase is the pulling apart of these sister chromosomes, and pulling to their respective nucleus. So this M phase makes sure that all the chromosomes are lined up so that the anaphase process can run smoothly. So CDKs are cyclin-dependent kinases, which depend on cyclin, and as you can see in the diagram, they bind together, and this bind is mostly important through the checkpoints because of the phosphorylating of materials, and it aids in the process of having cells go through the process. Two signals that provide stop and go signs at checkpoints. These are both internal and external. So MPF. So MPF is the combination of cyclin and CDK, as we saw in the beginning slide. And we know that the cyclin and CDK are responsible for the reasons why the cell can go past through the processes because of the phosphorylating materials. And the next signal is the PDGF. And the PDGF is mostly known for regulating the growth of a cell. And as you can see in the first slide to the right that popped up, the cells placed in the container, they have no PDGF. And you can see that growth is fairly slow. But as you see the other slide or picture that popped up, there's plus PDGF in it, and that's what aids and helps the cells grow. Because of their attached part, and cancer cells has differences from regular cells. It does not follow the cell cycle. It does not follow their cell checkpoints, and it's known to have their own, sort of like their own PDGF, to where they can grow and divide on their own. And this is a disruption from the cell cycle, as it just uncontrollably grows on its own, does not stop, and this is what leads to cancer. And this was my presentation. presentation.